So brain metastases, they are clinically devastating. They are the most common malignancy of the brain. They are one of the most feared complications of cancer. 25% of cancer patients will develop brain metastases, with the incidence rising as systemic therapies and extracranial disease control is improving. And historically, we've had a limited understanding of how brain metastases genetically evolve from their primary tumor. And why is this important to us as clinicians? Many patients progress only in the brain and not systemically. The question is, how different are brain metastases genetically from their primary tumors? When we make decisions about clinical trials and personalized medicine for brain metastases, can we rely on primary biopsies? We sought to further understand the evolutionary processes within brain metastases using massively parallel sequencing technologies. Our objectives were to, 1, elucidate the evolutionary patterns leading to brain metastases, 2, identify whether brain metastases harbor clinically significant genetic differences compared to their primary tumors, 3, determine the extent to which clinically significant mutations are shared among regionally, anatomically, and temporally distinct brain metastasis sites, and 4, determine whether lymph nodes or extracranial metastases are genetically similar to brain metastases and might serve as their proxy for clinical decision-making. We performed whole exome sequencing of 104 brain metastases matched with primary and normal tissue from the same patient. This included 15 with additional sites, so including extracranial metastases and additionally regionally, temporally, and anatomically distinct brain metastases. Notably, these were all biopsy samples collected as part of clinical care, and this is the largest massively parallel sequencing study of brain metastases and primary tumors to date. We developed novel computational tools to perform an integrative analysis of somatic mutations and copy number alterations. This analysis allowed us to estimate the clonal architecture of matched primary tumors and metastases, and to reconstruct a phylogenetic tree relating subclones from each patient. Across all our samples and phylogenetic trees, we find that the data is consistent with branched evolution, meaning we detect a common ancestor shared by the brain metastasis and the primary tumor, but there's further divergent evolution within the primary tumor and the brain metastasis. How is this important clinically? We next ask the question of whether brain metastases harbor clinically actionable mutations absent in their primary tumors. To answer this question, we annotated the phylogenetic branches of the trees from each matched case with clinically significant mutations or alterations that are associated with specific treatment opportunities in oncology. And we did in fact find that brain metastases are genetically distinct from their matched primary tumor biopsies. This is a representative example of a patient with a primary renal cell carcinoma diagnosed synchronously with the brain metastasis. The shared mutations are represented by the gray line here. Mutations exclusive to the brain metastasis represented by the red line, and mutations detected only in the primary tumor sample represented by the blue line. mTOR and VHL are shared mutations here. They are in the common ancestor of the primary tumor and the metastasis, with a clear driver mutation occurring in a hotspot of PIK3CA only in the brain metastasis. Furthermore, there was also loss of CDKN2A, again detected only in the brain metastasis, not in the clinically sampled primary tumor. When we look across all our samples, clinically actionable alterations were present on any branch of the phylogenetic tree. Here's a histogram of all clinically actionable alterations that occur in each case. Colors represent what branch they occurred on. Gray are shared by the primary and brain metastasis, red exclusive to the brain metastasis, blue exclusive to the primary tumor. Importantly, 53% of cases, more than half of cases, have a clinically actionable alteration in the brain metastasis branch, not detected in the primary site. The most common alterations were those in the CDK pathway, or alterations associated with sensitivity to CDK inhibitors. 51% of cases had alterations predicting sensitivity to CDK inhibitors. Mutations affecting the PI3 kinase pathway were also frequent. Actionable alterations in these genes occurred frequently in breast cancers, lung cancers, and renal cell cancers. So genetic divergence between primary and metastatic samples poses a major challenge to clinical decision-making for precision medicine and oncology in brain metastases. What about regional heterogeneity within the brain itself? How representative of bulk central nervous system disease is a single brain metastasis sample? To answer this question, we sequenced regionally, anatomically, and temporally distinct areas of brain metastases. And we found that clinically actionable mutations were shared even in metastases in regionally, anatomically, and temporally distinct areas of the brain in the same patient. This is a representative example of a patient with a salivary gland ductal carcinoma who had a symptomatic cerebellar metastasis resected pre-whole brain radiation and a parietal metastasis resected after whole brain radiation. And we sequenced two different regions of the cerebellar metastasis and the parietal metastasis. Both regions of the cerebellar metastasis and the parietal metastasis were more related to each other than either was to the primary tumor biopsy and shared the same clinically significant alterations not detected in the primary biopsy. Metastases within the central nervous system are relatively homogeneous. Given that central nervous system disease may be difficult to access in some cases, we then evaluated the extent to which involved regional lymph nodes and distal extracranial sites recapitulate genetic vulnerabilities present in CNS lesions. In this example case, we sequenced a primary colorectal cancer, a lung metastasis, and a brain metastasis. The brain metastasis had more alterations that were unique to the brain metastasis, as depicted by the long red branch here, and we also observed an amplification in MYC in the brain metastasis that was not present in the primary tumor. So brain metastases are genetically divergent from primary tumors and other extracranial sites. So we demonstrate divergent or branched evolution. How is this important clinically? If one were to exclusively sample the primary tumor or regional lymph nodes for selection of the appropriate targeted therapies, one may miss potentially actionable alterations in the brain. Our data show that clinically actionable variants occur in the metastasis branch 53% of the time, presenting a significant challenge to the application of precision medicine to brain metastases. So when a patient comes to our clinic, the question is whether we can make clinical decisions based on a single primary biopsy resected 5 or 10 years ago, and the answer is probably no. So in conclusion, every brain metastasis displayed branched evolution. Brain metastases harbored distinct clinically actionable genetic alterations compared to their primary tumors. All brain metastasis regions harbored the same actionable alterations, and extracranial metastases were not a reliable surrogate for brain metastases. And I'd like to acknowledge many individuals and institutions who have contributed to this work, namely Tracy Batchelor, David Lewis, Gadi Getz, and Scott Carter, along with many other institutions who've contributed samples and a lot of work to this work. And thank you again to our families, to our patients and their families.
Thank you very much.
Questions? Yes.
Ed Sussman with Oncology Times. Your study is fascinating, but it begs the question of, can you use this, have you used this clinically to drive treatment?
So the investigations to look at whether targeting the mutations based on what we see in the brain, they're underway now. So we're going to be investigating that now moving forward. So whether targeting the mutation in the brain will improve overall survival in these patients still remains to be unknown. I think what's very interesting in this study is that there was tremendous genetic heterogeneity in the brain metastases compared to the primary tumor, which was surprising. So, historically, it's been unclear if the differential responses we see in the brain and the primary tumor has been attributed to genetic heterogeneity, and this study suggests that this may, in fact, contribute to this differential response we see.
Lynn Peterson with Trends in Medicine. So you need to biopsy the brain mets?
So before we start advocating the biopsying the brain metastases, I think we need to do the study to show that targeting the brain metastasis, targeting what we see in the brain metastasis, will actually improve overall survival. So what we are suggesting is that so many brain metastases are clinically sampled. So patients undergo resections for solitary brain metastases, or they'll undergo resections for symptomatic brain metastases. If there is tissue available, we recommend using that tissue to look for targetable mutations. I think that's what we feel comfortable saying with this study. If that tissue is available clinically, as part of clinical care, then we should be looking for new targetable mutations in the brain. Whether targeting those mutations will improve overall survival, that's the next step to actually see if survival will improve. The other next step is obviously looking at liquid biopsy. So that's also what we are doing now at Mass General is looking for circulating tumor cells and circulating DNA to see if what we see circulating recapitulates what we see in the brain. Does that make sense?
So you're saying that in the liquid biopsy, can you tell where it's from? Can you tell whether it's from the brain met or the primary?
So that's exactly the issue with using liquid biopsies. We don't know, and we don't know if the liquid biopsy will represent what we see in the brain. We do not know if the liquid biopsy will represent what we see in the primary tumor. And we don't know if the mutations we see in the liquid biopsy are predictive of what we're going to see in the brain metastasis.
So if it's useful to biopsy the brain met to direct treatment, would that then mean there's a potential that we might not be able to move forward with liquid biopsies if they're only showing the primary tumor? So this has tremendous implications if I'm understanding this.
It has implications
Or
if that's the
biopsy.
case, if the liquid biopsies do not represent what we see in the brain metastasis. But that's a study that needs to be done, obviously.
But if it represented what's in the brain, then it wouldn't be able to represent both, would it?
So some of the mutations we found were shared. So some of the mutations were shared by both the primary and the brain metastasis, but the brain metastasis accumulated new mutations that were not present in the primary tumor. So whether the liquid biopsy is going to represent what we see in both, it still remains to be unknown.
How would you know which?
Well, the only way to do that is we're actually carrying out autopsy studies now at Mass General where we're actually comparing, we're looking at metastases from different sites as well as what we see circulating in these patients to see if what we see in the brain, what we see circulating represents what's seen in the brain. But that's a research study that's being carried out right now. But how you know whether it's what we see in the liquid biopsy is going to represent what we see in the brain, that still remains to be.
The brain or
the primary.
You don't know which without doing a regular biopsy.
Exactly.
Ever.
Right.
So basically, okay.
The genetic heterogeneity is daunting. I think that's what the study is showing. There's still a lot of work to be done.
Potentially big problem for liquid biopsy.
We don't know yet. We don't know. We'll have to do the study.
Hi, Linda Williams
Yes.
from Medwar News. I wanted to ask, from the patients that you looked at in this current study,
Yes.
were there any particular drugs that you felt that would have helped them because of the targeted mutations that you spotted? And if so, would they go past the blood-brain barrier, or would you have to deliver them directly into the brain?
Excellent question. So that's an ongoing study to see whether targeting what we see in the brain will actually lead to improved overall survivals. What we did find is that the brain did have these alterations, which in oncology, these alterations have been associated with treatment outcomes, such as the PI3 kinase mutations, such as PTEN loss. So these are mutations that traditionally in oncology have been associated with specific treatment outcomes in oncology. So we picked the genes based on what's used right now clinically and what's considered targetable. So, and the next question, whether these are agents that cross the blood-brain barrier, there are some agents that do, there are some others that don't. So this is an area of active research right now to find agents that cross the blood-brain barrier. So there are agents that are in clinical development right now. Great question. Yes?
So I'm still struggling with
Yes.
the liquid biopsy here, obviously. But if the drugs, you're talking about survival, but what if just reducing your brain mets is symptomatically better for the patient?
Mm-hmm.
So it doesn't necessarily have to improve survival.
Mm-hmm.
And what really matters is whether you can differentiate in your liquid biopsy between the primary and the brain.
Mm-hmm. Agreed. That's an excellent point. Agreed. And so that's what I think the next step is to So what we're doing now, so at this conference, I'm only presenting the results of this first data set, but we are sequencing hundreds of tumors now to actually see if there are any specific mutations that are predictive of brain metastases. And if we can identify those mutations that are predictive of brain metastases, then perhaps the liquid biopsy would be helpful in that setting. Does that make sense? So that remains to be seen. But I agree, if we were able to target the brain metastases, that could lead to improved outcomes in the sense that many patients do have significant side effects or significant toxicities from brain metastases. So a lot of symptoms from brain metastases, and a large portion of patients. So it's variable what you see in the studies, but some studies have suggested that up to 50% of patients with symptomatic brain metastases will die of their brain metastases. So by shrinking the brain metastases, one would hope that that could improve patient outcomes. Does that make sense? Okay.
Did the patients, had they undergone radiotherapy for the brain mets? Because that can change the mutational profile as well.
Some of the patients had undergone radiation therapy, and we actually compared the patients that had undergone radiation therapy versus not to see if the evolutionary patterns were different or if we saw any different mutations in those patients, and we didn't see any differences in those patients. However, again, this was a very heterogeneous study. Now, as we're moving forward and sequencing more tumors, and more homogeneous patient populations, we may be able to answer that question more fully.